Hi students, in last few lectures we have studied inheritance and variation. We have studied basic about genetics, then various terms used in study of genetics, then Mendel's work, characteristics of pea plant, then the crosses carried out by Mendel, that is monohybrid cross, dihybrid cross, and the principles or the laws derived from these crosses, that is law of dominance, law of segregation, and law of independence assortment. Today we have to study another important cross carried out by Mendel, and that cross is back cross. That cross is back cross. Study back cross. Now what is back cross? First we study the definition. Back cross, it is the cross. Back cross is the cross between F1 individual and any one of its parent. When F1 individual is crossed with any one of its parent, then it is termed as back cross. Okay. Example. Suppose the first cross, that is the cross carried out by Mendel, that is monohybrid cross. In monohybrid cross, we have studied the phenotype, then its genotype, then gametes, and F1 generation. Phenotype is tall plants, tall plant crossed with dwarf plant. Tall plant crossed with dwarf plant. These two plants are homozytes. The parents are homozytes. So the genotype is capital T, here small t, small t. Okay. Gametes, homozygous parents, and hence only one type of gametes, it is capital T, and here small t. In F1 generation, there is formation of new individuals, and these individuals are heterozygous, that is hybrid tall plants. All individuals in F1 generation are indeed hybrid tall. What is back cross? It is the cross between F1 generation individual with any one of its parent, and hence this back cross is of two types. Back cross is of two types. That is, if the used parent is dominant, then it is termed as dominant back cross. Dominant back cross. And when F1 individual is crossed with the recessive parent, then it is termed as recessive back cross. So we have to study these two crosses one by one, that is dominant back cross and recessive back cross. So the first back cross, that is dominant back cross. We have to study what happens in such crosses and what is the importance of these crosses in the study of genetics. Okay. So number one is dominant back cross. Dominant back cross. What is dominant back cross? What is back cross? Back cross is the cross between F1 individual and any one of its parents. Here, when F1 individual is crossed with dominant parent, then it is termed as dominant back cross. So first we study the such dominant back cross with the example of monohybrid cross. In monohybrid cross, example, tall pea plant crossed with dwarf pea plant. Then in F1 generation, all hybrid tall plants, that is capital T and small t. Now we have to cross this F1 individual with dominant parent. So F1 individual crossed with dominant parent. Here the genotype of F1 and phenotype of F1. Now what is the phenotype of F1 individual? It is tall and dominant parent, obviously it is also tall. But here genotype is different. Here F1 generation genotype is capital T, small t. Here genotype is capital T, capital T. Okay. Heterozygous, homozygous. This heterozygous individual forms two types of gametes, that is capital T and small t. Okay. And this parent forms only one type of gametes, that is capital T. Random fusion, capital T, and here capital T, small t. Okay. Phenotype, hybrid tall is also hybrid tall. Okay. Now, what is the importance of such cross? Which cross? Dominant back cross. First, listen carefully. F1 crossed with dominant parent, that is the hybrid tall plant crossed with homozygous tall plants. Then the result is all the individuals formed in such crosses are with only dominant character. It is the all plants are tall plants. All plants are tall plants. Okay, remember this. Now the second back cross is more important because it has very important significance in plant breeding. So the second one is a recessive back cross. A recessive back cross. Now what is recessive back cross? It is back cross, that is F1 individual crossed with its recessive parent. Okay. recessive backcross, F1 crossed with a recessive parent. Now here in monohybrid cross, F1 individual is with genotype capital T, small t. Recessive parent, small t, small t. Okay. Here two types of gametes, capital T and small t. And here small t. Random fusion, capital T, small t. Here small t, small t. This plant is tall. This plant is dwarf. Okay. In dominant backcross, all the individuals shows only dominant character. That is the expression of only dominant allele. While in a recessive backcross, the individuals formed are in 1:1 ratio. That is 1 tall and 1 dwarf, or 50% tall and 50% dwarf. Okay. Such cross, that is the recessive backcross, that is the cross between F1 individual and a recessive parent, this cross is termed as test cross. Is termed as test cross. Understand carefully. Okay. Dominant backcross is not the test cross. Dominant backcross is not the test cross, but the recessive backcross is the test cross. Okay. All backcrosses and all test crosses. Okay. Dominant backcross is backcross, but recessive backcross is termed as test cross. Okay. This test cross, F1 individual crossed with recessive parent. In board examination, many times this question was asked. That is, what is test cross? Test cross is a cross between F1 individual and its recessive parent. And what is the result of test cross? Result of test cross is ratio is 1:1. That is 50% tall and 50% dwarf. Means 50% with dominant character and 50% with recessive character. But these 50% tall plants are heterozygous, not homozygous. These plants are heterozygous. Oh, okay. It is the example of monohybrid recessive backcross. It is what is it? It is monohybrid test cross. Monohybrid test cross. It is monohybrid test cross. Now we have to study same test cross with dihybrid cross. Okay. With dihybrid cross. First we see the example of dihybrid cross, how one individual appears, and then we have to study the recessive backcross along with dihybrid cross. Okay, we have studied one example in previous lecture, that is the dihybrid cross. Example here, phenotype, then genotype, gametes, and you have one generation. Suppose one example, yellow and a round seeded pea plant crossed with green and a wrinkled seeded pea plant. Okay. Yellow and round seeded pea plant is crossed with green and wrinkled seeded pea plant. Okay. Here phenotype is, remember this example, yellow and round seeded pea plant crossed with green and wrinkled seeded pea plant. Phenotype is yellow and round, yellow and round crossed with green and wrinkled, green and wrinkled seeded pea plant. Here capital Y, capital Y, capital R, small y, small y, small r, small r. Gametes, capital Y, capital R, here small y, small r. F1 generation, capital Y, small y, capital R, small r. It is dihybrid. Remember, it is dihybrid with yellow and round seeds. It is dihybrid with yellow and round seeds. Now remember this F1, dihybrid yellow and round seeded pea plant with genotype capital Y, small y, capital R, small r. We have to study recessive backcross with dihybrid crops. Now actually recessive backcross. In recessive backcross, again the phenotype is yellow and round. It is F1 crossed with a recessive parent. Crossed with recessive parent. Yellow and round seeded pea plant crossed with green and wrinkled seeded pea plant. Green and wrinkled. Here the genotype of F1 individual is capital Y, small y, capital R, small r. Here small y, small y, small r, small r. Then the gametes. Here four types of gametes, that is capital Y, capital R, capital Y, small y, capital R, and last small y, small r. And here small y, small r. Then the fusion. What happens in F1 generation or the next generation? What happens? Here it goes with capital Y, small y, capital R, small r. It goes with capital Y, small r, small r. Then small y, capital R, small r. And last one is small y, small r, small r. Okay. Here four types of offsprings are formed. One is with yellow and round seeds. It is yellow and wrinkled seeds. It is with green and round seeds and green and wrinkled seeds, and hence the ratio is 1:1:1:1. Remember, in monohybrid test cross ratio is 1:1. In dihybrid test cross ratio is 1:1:1:1. Okay, so it is the back cross, test cross, monohybrid test cross and dihybrid test cross. Now last important concept. What is the importance of this test cross? One most important significance of this test cross is this cross is used to identify the genotype of unknown parents. Remember, this cross is used to identify the genotype of unknown parents, and due to its simple ratio, it is used in plant breeding for improvement of the crop plants or to introduce the desirable recessive character in next generation. Okay, thank you.